Hey everybody. Okay, I'm going to try this out using the webcam to do a little teaching on the board. I know I'm blocking the board right now, but I want to make sure you are able to see everything. Okay, so we're going to do a little review about what we were learning before we went on this nice long break. And that is, if you remember, we talked about DNA replication. Hopefully this looks familiar. I'm going to tilt it this way a little bit. And just to recap, with DNA replication, when the cell needs another DNA molecule before it divides, the DNA molecule will replicate. So if you remember, we have an enzyme called helicase, and helicase comes through and separates the two strands of DNA. Once the two strands of DNA are separated, then the enzyme DNA polymerase came through, and it put those matching nucleotides, A with T, C's with G's, apples in the trees, cars in the garage, remember that? They go ahead and pair up. They pair up on each of the single strands that's separated. And when we're all done, we have two identical DNA strands. They are similar to each other. They're identical to the one in which they came from. And if you remember, the cool thing is that every new DNA strand is half old, or comes from the original, and half new. Brand new strand from the free nucleotides that came in and paired up. I'm showing you that because transcription, which is the new topic we have to talk about today, is very similar to DNA replication. But in transcription, move you over so you can see transcription. In transcription, we are going to be making an mRNA molecule. Not a DNA molecule, but an mRNA molecule. So hopefully at this point you remember that the mRNA molecule is the molecule that carries instructions on how a protein should be made. We don't want the cursor on my face. All right, so in regards to how transcription takes place, it does take place in the nucleus, just like DNA replication. And there is an enzyme that comes through and separates the two strands of DNA. Just like in DNA replication, they are going to separate. So one strand will be on one side, and the other strand will be on the other side. Then there's going to be an enzyme that comes through. This enzyme is going to be called RNA polymerase. Kind of appropriate, since we're trying to make an mRNA molecule, that the enzyme that will help make that mRNA molecule is called RNA polymerase. But if you remember, mRNA is single-stranded. It's not double-stranded like DNA. So that means only one of these two strands of DNA are needed to make our mRNA copy. So we're going to use this strand. And so as we're going through and using this strand here, we're going to put in the matching nucleotides. But these nucleotides are going to be RNA nucleotides. And if you remember, they're a little different than DNA nucleotides because they have the sugar ribose instead of deoxyribose. And they're going to use the base uracil instead of thymine. So right away, normally we'd say A would pair with T if we were making another DNA strand. But we're not. We're making mRNA. So A's are actually going to pair with U's because RNA uses uracil instead of thymine. C's and G's are the same. C's go with G's, C's go with G's. T's, even though they're not going to be in our RNA strand, still can match with A's because mRNA does have A's. C's with G's. And now here's an example again where A is going to go with U, not T. So you have the U here. This then becomes our mRNA strand. And what will happen is the mRNA then will leave the nucleus, go find a ribosome, and we'll say, ribosome, build this protein. Because to the cell, again, this is instructions for building a protein. The ribosome will be able to read it, bring the correct amino acids that are needed to build the protein. Then, once mRNA leaves the nucleus, if you remember from back in chapter 7, we were looking at the organelles. The nucleus, if you remember, has nuclear pores, little tiny openings. That's how mRNA gets out of there. So it leaves through nuclear pores. And once it does, the two strands of DNA, they actually come back together again. their hydrogen bonds form and they connect it together again, and they wait until the next time the cell needs to carry out that set of instructions. Okay, so again, that is transcription, the making of an mRNA molecule. So the cell has the instructions on how to go ahead and create the protein. Now, the way I showed you transcription here is showing the 2 strands of DNA separating, 1 strand is being used to make the mRNA. But next week, when we do some work with decoding DNA into mRNA and then looking up the names of amino acids to figure out the proteins, you're going to see it in a little bit different format, which is what you see over here. So we're going to travel over there. Here comes the computer. Okay. We can do better. Okay. So sometimes you're going to be asked to create an mRNA molecule from a single strand of DNA. You're going to do the exact same thing we do over here. The only difference is instead of having DNA be double-stranded and having to separate it out first and pick 1 strand to be used as our template, I'm just going to give you a single strand that I want you to use as the template. Okay, so it's actually easier. It eliminates the work. So instead of having the strand going vertical, the strand is just horizontal. But everything else is the same. So if I wanted to make an mRNA from this DNA strand, A's would pair with good, U's, C's would pair with G's, T's still go with A's because RNA has A's, G's go with C's, and A's again are going to go with U's. Okay, so I'll be asking you to do this sometime next week, whether that be in person or back here online. And then we'll decode them to figure out which amino acids will be brought and which protein would be made. So again, that is transcription, the making of an mRNA molecule, so that the cell then can get the information that it needs to build a protein. As always, please reach out to me if you have questions, and if this doesn't make any sense, let me know so we can go through it together one-on-one on Hangouts or anything like that. So again, stay healthy. Hopefully you're enjoying this new experience of online, and hope to see you guys soon sometime, whether it be over the computer or in person when we're all healthy. See ya!